Hello dear students, this is Dr. Vishwajit Ghorpade, and in today's video lecture, we are going to talk about structure of DNA. The learning objectives of today's video lecture are, after completion of this video, you will be able to describe the structure of DNA, you will be able to identify the types of DNA, and you will be able to state the functions of DNA. So let us begin with our today's topic. We all know that long form of this DNA is deoxyribonucleic acid. We call it deoxyribonucleic acid because it contains deoxyribose sugar in it. It is a polymer of deoxyribonucleotides. Means the deoxyribonucleotides which we have seen in our previous lecture, these deoxyribonucleotides are going to act as monomer units for this DNA. And such many monomer units, such many deoxyribonucleotides get connected to each other to finally form the DNA. Which are these monomeric units which constitute the DNA? There are 4 monomeric units. There are 4 types of deoxyribonucleotides which usually constitute this DNA. Which are those 4 types? They are given below. The first monomeric unit or the first deoxyribonucleotide is deoxyadenylate, that is DAMP, where small d indicates deoxyribose, AMP is adenosine 5-monophosphate. Second monomeric unit or deoxyribonucleotide is deoxyguanylate, which is indicated by small d GMP, that is guanosine 5-monophosphate. The third monomeric unit is deoxycytidylate, that is DCMP. And last monomeric unit or deoxyribonucleotide is deoxythymidylate, which is indicated by small d TMP, where TMP indicates thymidine 5-monophosphate. These 4 monomeric units or deoxyribonucleotides are usually linked to each other to form polydeoxyribonucleotide segment. And this polydeoxyribonucleotide segment constitutes the strands of DNA. Here you can observe one polydeoxyribonucleotide segment. So the diagram of that polydeoxyribonucleotide segment has been shown over here in the middle. Here you will see that various nucleotides, for example, you can see that here this nucleotide is nothing but it is AMP. The middle nucleotide is GMP and the last nucleotide is TMP over here. And these nucleotides are linked to each other or connected to each other by phosphodiester bond. So this is the phosphodiester bond. Why we are calling it as a phosphodiester bond? If you consider the diagram shown towards the right hand side over here, it can easily help you to understand what is a phosphate diester. Now you can see over here that this is the image of phosphate diester. We are calling it as a diester because there are 2 ester groups over here. The first ester group is over here and second ester group is over here. So as 2 esters are involved in this structure, we are going to call it as a phosphate diester. Only the difference is that instead of carbon, there is phosphate over here in the middle, and that is why we are going to call it as a phosphate diester. Okay. Now, whenever there is a phosphate diester linkage, that linkage is usually formed in between C3- carbon of deoxyribose sugar of one nucleotide and C5- carbon of deoxyribose sugar of another nucleotide. So finally we are going to get this structure over here. You can easily see the same thing over here which I have told you just now. Let us consider the linking in between AMP and GMP. You will see that the phosphodiester linkage is formed in between C3- carbon of deoxyribose sugar of this nucleotide and C5- carbon of deoxyribose sugar of this nucleotide. Now, this polydeoxyribonucleotide segment is going to form single strand of a DNA. Now, usually to easily draw this polydeoxyribonucleotide segment, we prefer a shorthand structure of this polydeoxyribonucleotide segment. The shorthand structure consists of simplified diagram which will help you to understand the structure of polydeoxyribonucleotide segment or simply the structure of DNA. So let us see what is that shorthand structure of polydeoxyribonucleotide segment. So towards right hand side, you can see the shorthand structure of polydeoxyribonucleotide. Here you will see there are three parallel horizontal lines. These three parallel horizontal lines indicate the deoxyribonucleic sugars which are involved in these three deoxyribonucleotides. Now concentrate on this first horizontal line. This first horizontal line indicates this deoxyribonucleic sugar over here. Okay, where you will see that at the left hand side end of this line is C5-phosphate linkage. In middle there is C3-phosphate linkage, and towards right hand side there is C1-carbon which is usually attached to adenine. Similarly, the things can be predicted for the other two nucleotides over here. Now let us see what is the Chargaff's rule of DNA composition. In 1940, Irving Chargaff, he found that DNA has equal number of adenine and thymine residues. That is, whatever will be the number of adenine residues in the DNA, exactly that much number of thymine residues will be present in DNA. Similarly, he also found that the number of guanine and cytosine residues are also equal. Means whatever will be the number of guanine residues, exactly that will be the number of cytosine residues. And hence, finally it was proved by Chargaff that there is a molar equivalence in between purines and pyrimidines in DNA. Keep one thing in mind, that in double-stranded DNA or double helical structure of DNA, whatever ladder structure we observe, that ladder structure involves the pairing of purines with pyrimidines. And whatever will be the number of purines, exactly that much will be the number of pyrimidines. And hence we can say that there is molar equivalence in between purines and pyrimidines. Means whatever will be the number of purine residues, exactly that much will be the number of pyrimidine residues. However, this rule is not obeyed by single-stranded DNA and RNA. Means it is not applicable to the single-stranded DNA and RNA, which is also single-stranded. It is only applicable to double-stranded nucleic acids. Now here, towards right-hand side, you can observe the double helix structure of DNA. This double helix structure of DNA was proposed by James Watson and Francis Crick in 1953, for which they have received the Nobel Prize. Let us see what are the salient features of this double helix structure of DNA. Firstly, it is a right-handed double helix structure. It is a right-handed double helix structure means here whatever twisting happens in between the two strands of DNA, it happens in clockwise direction. Means you will see that here are two strands of DNA which are twisted around each other, and while twisting they are having a common rotation axis, and this twisting happens in clockwise direction along right hand side. If you have still not understood how this twisting happens in clockwise direction, you can consider this image over here. So this is the spiral staircase which is twisted in clockwise direction. Means if you imagine yourself that you are coming down along this staircase, then you will find that you are moving in clockwise direction. Similarly, in DNA also, whatever twisting takes place in between two strands, it happens in clockwise direction. Okay, so here you will see that twisting in the staircase is in clockwise direction. Means when you are going to come down along the staircase, you are going to move in clockwise direction, and then only you are going to come on this floor. The second thing is that the two deoxypolyribonucleotide are twisted around each other. So here you can observe that there are two strands, two polydeoxyribonucleotide segments which are twisted around each other. The first segment is shown in pink color over here, and second segment is shown in black color over here. Third salient feature is that the strands usually run in anti-parallel direction. Okay, means whichever two strands you are observing over here, one in pink color and one in black color, they will always run in anti-parallel direction. If one strand is running from 3' to 5' end, the another strand will run from 5' to 3' end. So they are running in anti-parallel direction. Also, you will see that in this twisted structure of DNA, there are certain grooves present. Okay, so here you will see that there is a groove present, but this groove is a minor groove. Okay, it is a smaller groove over here. The depth is small. But if you observe this groove over here, this groove is a larger groove. It is a major groove because its depth is more. Now, what is the significance of this minor groove and major groove? Whenever the proteins are going to interact with this DNA, the proteins interact along this major and minor groove over here. Okay, so proteins are going to interact with DNA at this minor groove and this major groove only. They are not going to disturb the base pairing over here in between. They will be interacting only at this major and minor groove. Now, if we consider the width of this DNA, okay, so if you consider the width over here, look towards right hand side, towards the pointer which I am showing, the width of this DNA is 2 nanometer. Okay, it is also indicated by 20 Armstrong. Keep one thing in mind that 1 nanometer is 10 Armstrong. That is why if the width is 2 nanometer in Armstrong, it will be 20 Armstrong, which is shown over here. Secondly, if you consider the length of each turn, okay, so if you consider length of each turn over here, it begins from this point over here and it ends at this point. So this is one complete turn. It begins from here and ends over here. So this is the one complete turn. And if you consider this one complete turn of this double helical structure of DNA, the length of this one single turn will be 3.4 nanometer, or simply it will be 34 Armstrong. In this one single turn, there are total 10 pairs of nucleotides. Here, if you count the number of nucleotides, you can count simply these horizontal lines present in between these two strands. You will see that there are 1, 2, 3, 4, 5, 6, 7, 8, 9, and one nucleotide will be present over here in between this twist. It will not be observed because it will be present over here in between this black and red strand in middle, and that is why it is not observed over here. So total there will be 10 pairs of nucleotides in one complete turn of DNA. Thereafter, the distance in between each pair, if you consider the distance in between each pair, each base pair or each nucleotide over here, that distance is usually 0.34 nanometer, or you can call it as 3.4 Armstrong. Now, the backbone of DNA is nothing but it is the strand of this DNA, and this strand is mainly made up of 3-5- phosphodiester. So backbone of DNA is nothing but it is the strand of this DNA, and it is made up of 3-5- phosphodiester bond. Whereas the inner core of this DNA is made up of bases. You can easily find that in between whatever ladders you are observing in this DNA, those ladders are made up of the nitrogenous bases over here. So the inner core is formed of bases. That is what we can say. Now, the polynucleotide chains which we observed in DNA, the polynucleotide chains, or we can say the strands which we are observing in DNA, these strands are usually complementary to each other. They are not similar to each other. They are complementary. Means one strand will fulfill the needs of another strand. Okay. That is why it is usually said that polynucleotide chains are complementary to each other, because they are fulfilling each other's needs. Now, what needs they are fulfilling of each other? See that whenever the base pairing occurs in between the two strands of DNA, at that time, adenine, which is a purine, will pair with thymine, which is a pyrimidine. Similarly, guanine, which is a purine, will pair with cytosine, which is a pyrimidine. Now, if this strand wants to pair with another strand, at that time, it should be considered that if this strand is containing adenine at this point, then the other strand must contain thymine on the other side. Similarly, if this strand contains guanine at this point, then the other strand must have cytosine on opposite side. then and then only the pairing will happen, then only a double helical structure of DNA will get formed, and hence it is said that polynucleotide chains are complementary to each other. The another salient feature is that the two strands of DNA are held together by hydrogen bonds. Now see, this strand and this strand are connected to each other by hydrogen bond. Means this hydrogen bonding, it exists in between the bases of each of these strands. So if you see over here, the adenine is linked to thymine, and here it is linked to thymine by two hydrogen bonds. These two parallel lines which you are observing over here, they indicate the two hydrogen bonds. Similarly, you will see that there are three parallel lines in between guanine and cytosine. This means there are three hydrogen bonds existing in between guanine and cytosine. And in this way, one strand of DNA will be connected to another strand of DNA with the help of these hydrogen bonds. And keep one thing in mind, that purines will bind with pyrimidines only. So whichever purines are there, they will bind with pyrimidines. Now in this image, you can observe that how the hydrogen bonding happens in between the bases. You will see that in between adenine and thymine, there are two hydrogen bonds over here. That hydrogen bond is formed in between the hydrogen attached to this amino group at 6th position of this purine parent ring and carbonyl oxygen of this thymine. Similarly, the hydrogen attached to this nitrogen of thymine will form a hydrogen bond with this nitrogen which is present at 1 position in the purine ring structure of adenine. So there are two hydrogen bonds which are formed in between adenine and thymine. Similarly, if you consider the bonding in between cytosine and guanine, you will observe that there are three hydrogen bonds present in between cytosine and guanine. Now, the genetic information, it resides on the sense strand. If we consider these two strands of DNA, among these two strands of DNA, one strand is going to be a sense strand and another strand is going to be a antisense strand. Antisense strand, it helps in formation of RNA, which will further lead to synthesis of proteins. Whereas the sense strand is going to contain the genetic information which will get passed from one generation to another generation. Now there are 6 forms of DNA: A, B, C, D, E and Z. Okay, and these types are having different conformations. Among these 6 types, 3 types are important, that is A, B and Z. Now what is the difference in between A type, B type and Z type? Let us see. So here in this table, you can easily find out what is the difference in between B DNA, A DNA and Z DNA. Firstly, if you consider the type of helical structure of these 3 DNA, B DNA has a right-handed helical structure, A DNA also has a right-handed helical structure, whereas Z DNA has a left-handed helical structure. Means here the twisting will happen in anti-clockwise direction, whereas in first 2 cases the twisting will happen in clockwise direction. The diameter of the helix is 2.37 nanometer in B DNA, it is 2.55 nanometer in A DNA and it is 1.84 nanometer in Z DNA. If you consider the distance of one complete turn of DNA, then in B DNA it is 3.4 nanometer, A DNA is 3.2 nanometer and Z DNA is 4.5 nanometer. If you consider the distance in between 2 nucleotides of DNA, then in B DNA it is 0.34 nanometer, in A DNA it is 0.29 nanometer, in Z DNA it is 0.37 nanometer. And number of bases in one complete turn of DNA, in B DNA it will be 10 base pairs per complete turn, in A DNA it will be 11 base pairs per complete turn, and in Z DNA it will be 12 base pairs per complete turn. Now, if you properly compare these 3 DNAs, you can easily predict that the DNA which we have studied up till now was B DNA. Now, let us talk about size of DNA molecule. The molecular weight of B DNA is 660 Daltons. Okay, And whenever we want to measure the length of this double-stranded DNA, that length is usually measured in terms of base pair. It is the unit of measuring the length of DNA. And of course, it should be a double-stranded DNA. So this base pair is indicated by abbreviation BP. So whenever we are going to measure the length of double-stranded DNA, we are going to measure it in terms of base pairs, that is BP. Now these base pairs can also have higher values, and those higher values are measured as kilobase pair, megabase pair and gigabase pair. The conversion of kilobase pair to base pair, megabase pair to base pair and gigabase pair to base pair is given over here. So 1 kilobase pair will be equal to 1000 base pair, 1 megabase pair will be equal to 10 raised to 6 base pair, 1 gigabase pair will be equal to 10 raised to 9 base pair. If you want to measure the length of single-stranded nucleic acid, you cannot use the base pair unit to measure its length, because base pair unit is only used to measure the length of double-stranded DNA. If you want to measure the length of single-stranded nucleic acid such as single-stranded DNA or single-stranded RNA, you cannot use this measure to measure the length of single-stranded DNA or RNA. The length of this DNA can also be expressed as contour length, means along with base pair, we can also measure the length of DNA as contour length. What is this contour length? It is the total length of genomic DNA in the cell. Means whatever will be the total length of genomic DNA in the cell, that total length will be measured in terms of contour length. This contour length of DNA in human diploid cell is 2 meters. So in a human diploid cell, if you measure the contour length of DNA, then that contour length of DNA is 2 meters. So it is very big as compared to the size of cell or the nucleus. As this size is greater than the nucleus, now see the size of nucleus is 10 micrometers and the contour length of DNA is 2 meters. In order to fit this 2 meter length of DNA in 10 micrometer size of nucleus, the DNA is usually arranged in a compact manner. Now, what are the functions of DNA? First thing is that the DNA acts as a reserve bank of genetic information. This means that DNA is going to act as a storehouse of genetic information, and hence it is going to help to pass the genetic information from one generation to another generation. The second function is that it maintains the identity of different species of organisms over millions of years. In archaeology, whenever certain fossils are observed, from the DNA of that fossil, it can be identified, that is, to which species that fossil belongs to. And hence, we can say that DNA can help to identify different species or organisms which are buried in the ground and are found in archaeology over millions of years afterwards. The third function is that it controls every aspect of cellular function. This is because DNA helps in the formation of RNA, RNA helps in the formation of proteins, and these proteins are very helpful for various cellular functions. And hence we can say that DNA controls every aspect of cellular functions. So, this is the reference which I have used for this video lecture. I hope that you have understood the structure of DNA from this lecture. Thank you.